going to learn about how does DNA encode information? How does DNA replication ensure genetic constancy during cell division? And the last one is how do mutations occur? So this is the 5 main goals that we are going to discuss. Yeah.
Yes.
So the first one, how did scientists discover that genes are made of DNA? Yeah. So by the late 1800s, yeah, scientists had a limited knowledge of genes. Yeah. So the heritable information is carried in discrete units called genes. Yeah, we already give genes definition, but they did not know what genes were made of. Yeah. So if you see here that genes are parts of structure called chromosome, chromosome are made of deoxyribonucleic acid or DNA and protein. This invention, this discovery came later on. And then the next one. Nah, how we found that there is a link between genes and DNA is by this transformation experiment conducted in bacteria. Yeah, in the 1920s, Frederick Griffith worked with 2 strains of Streptococcus pneumoniae bacteria. You don't need to memorize the later name, do not worry. But in this part, what you need to know is the main pointers of the experiment. Yeah, so the main idea is the big picture. So there are several strain. We use 2 strain, yeah, the S strain and R strain. The S strain is the strain that can cause pneumonia when injected into mice. Yeah, so we inject the S strain into mice, and then when we examine the mice, yeah, they will get infected with pneumonia. Meanwhile, the other strain called the R strain, this one did not cause pneumonia when injected. So when any mice injected by this R strain, yeah, it will it will not get infected with pneumonia. Now, what happened when we killed the S strain? For example, we heat up the bacteria colony, yeah, and then we inject it into the mice. We know when we already killed the bacteria will become inactivated, so it cannot cause disease anymore. Yeah, in other words, it loses its capability to cause diseases in an organism. Now, how Griffith made the transformation experiment is by made a sample of heat-killed strain. So you have the strain, right? The previous one. Let me draw it on the board here. This one. So you have the strain, this one, in red. Yeah. And then I heat it up like this one. So it is now inactivated, right? Cannot cause pneumonia. And then I mix it with the R strain. R strain, I will give it as a blue dots like this one. Yeah. Now, when I mix those 2 colonies of bacteria, yeah, the injection of this combination into the mice. So this is the mice. Yeah. This combination will cause pneumonia and death. This was unexpected since neither element alone caused the disease. Yeah. So you know if it is R strain, it is not causing the disease. S strain that is already heated also cannot cause the disease. But when you mix both type, these bacteria can undergo a sexual reproduction process called as transformation. Nah, So this is answering our question on how comes the mice get infected, yeah, Leah, by this kind of treatment because it is due to this transformation process, yeah, the sexual reproduction process, yeah, that happens on the between the two colonies. Later on, I'll send you the pictures also. Any question up to this point?
No.
Okay, now I'll go into the detail. Yeah, the results of Griffith experiment. This is how scientists work. Yeah, to discover new invention in science. Yeah, they made new deduction about the genetic material. Oh, we know that there is some substance in the heat-killed S strain that changed the living harmless R strain bacteria into the deadly S strain, a process Griffith called as transformation, right? The substance that caused this transformation might be the long sought molecule of heredity. Yeah. So we already found the candidate, yeah, candidate molecule. Now this is the process in pictures. So if you see there are 4 rows, start from row A, living R strain. And then you injected it, the results, most remains healthy. The conclusion is that our strain does not cause pneumonia. And then at the second row, sorry, you injected living S strain into the mouse. And then the mouse contract pneumonia and die. Conclusion, S strain caused pneumonia. And then heat killed S strain, obvious because you heat it up, the bacteria lost its pathogenicity. Therefore, the mouse remains healthy. What is the conclusions? Heat-killed S strain does not cause pneumonia. Right. And then the last one. Mixture of living R strain and heat-killed S strain. Yeah, what is the result? Mouse contract pneumonia and dies. This is what we talked about in the previous slide. The conclusion is a substance from heat-killed S strain can transform the harmless R strain into a deadly S strain. So it is like a science fiction, yeah, a bit of science fiction movie, yeah, when the dead one can influence the living one to become a zombie, yeah. So that's the scenario. How come? How it can happen? In 1933, yeah, conducted by another scientist. So you see that the body of knowledge in science is rarely the work of one person only, yeah, because we work together in science to make amazing discovery. Yeah, this one, the Griffith experiment continued by another scientist, G.L. Alloway, showed that transformation occurred just as readily in cultured dishes. This showed that the mouse in Griffith's experiment was not involved in the transformation. So the transformation does not involving the animal, but something that is outside of the animal body. What is it? The microorganism, yeah, the cell of the bacteria. Nah, the transforming molecule is DNA. How come? Another scientist again come to the pictures. In the 1940s, yeah, there are 3 scientists, Oswald Avery, Colin MacLeod, and Maclean McCarty, isolated DNA from S strain bacteria and then mixed it with live R strain bacteria and produced live S strain bacteria. This suggested that the transforming molecule from the S strain was DNA. Yeah, because now this scientist wants to prove, oh, I want to prove the hypothesis, yeah, the presumption that DNA molecule is the one who is causing all of this phenomenon. So what I should do then? I isolate the DNA first. So isolate means, yeah, I just take the DNA and separate the DNA from the other content of the cell. Because in cell it is a mixture, right? You have carbohydrate, you have fats, and another type of molecule. So I isolate this one, just the DNA, yeah, to rule out the possibility that a protein contaminant was actually causing the transformation. Remember, you have carbohydrate, you have protein, and if you also suspect protein, you want to rule out that possibility, that suspicion, yeah. So Avery also treated the samples with protein destroying enzymes. Yeah, protein destroying enzymes. And then he still observed that induced transformation still occurred. So it means it is not protein. Why? Because you already destroyed the protein using enzyme and still transformation can take place. So it means it is something other than protein. Okay. Now, and then one more key step to answering the $1 million question. They add DNA destroying enzyme, yeah, into the samples. And when they add that, they observe it, they found that transformation did not occur. Yeah. So they can conclude that the transforming molecule is DNA. Yeah.
A question.
Yes.
What was enzyme?
Now, what was enzyme? That is a very good question. So, enzyme is a biological catalyst. It is a chemical that can speeds up the rates of a reaction. So, you already learned chemistry. Remind a bit about chemistry. In chemistry, we have chemical reaction, right? Like rusting and then the spoiling of an egg. That is the chemical changes. Yeah. Now, during those processes, some enzyme may involve in the process. Why? To speed up the rate of reaction. Without enzyme, chemical reaction still can take place, but it takes a very, very, very long time. For example, a certain chemical reaction, yeah, without enzyme, it can finish up until 1 million year, 2000 years, very long. If you just rely on spontaneous mechanism on how they react. But when you use enzyme, yeah, they will occur in a much, much faster rate. And a lot of enzyme can be encountered in a body of an organism. Either it is bacteria, fungi, animal, plant, human, all of us has this molecule called enzyme because of the role that is so vital, that is so important for the processes of life. We know that life is a process that is very dynamic and the chemical reaction should occur very fast. Yeah, life cannot happen in the first place if we do not have the enzyme. Okay?
Yes.
Nah, okay, thank you, Leah. Now, I'll continue, yeah, to the next part. Yeah. So Griffith experiments can be explained if DNA is the transforming agent. Because, one, heating S-strain cells killed them, but did not completely destroy their DNA. Yeah. So when you heat up a microorganism, you may kill the microorganism, but the molecules that constitutes that organism is still there. And if you heat it up until a certain point only, not destroying all of the components, it is not hot enough to destroy all. It means that you still left some of the DNA molecule intact. So when the killed S-strain bacteria were mixed with living R-strain bacteria, fragments of DNA from the dead S-strain cells become incorporated into the chromosome of the R-strain bacteria. If these fragments of DNA contain the genes needed to cause disease, an R-strain cell would be transformed into an S-strain cell. Thus, Avery, MacLeod, and McCarty concluded that genes are made of DNA. Yeah. So these three scientists concluded that genes are made of DNA. Yeah. Because the R strain, yeah, will take those DNA. And then this DNA from the dead will become incorporated into the chromosome of the R strain bacteria. So remember the zombie story, yeah, to make it easier. Now this is happening, yeah, but this is in bacteria, yeah. We call it as the horizontal gene transfer, yeah, horizontal gene transfer. Later on, how about from plant to insect? There is some, yeah, some plants that can transfer its genes to the insect also, but that is for another discussion. Right now, you just need to know that this phenomenon occur in bacteria. Yeah. This one. Molecular mechanism of transformation. So you see the yellow circle here, yeah. We call it as bacterial chromosome. And then you have foreign DNA fragments in blue that can be transported into the bacterium. Yeah, the DNA fragments can be transported into the bacterium. Yeah, because bacteria has certain transporter. So transporter is basically a molecular machine that permits the foreign DNA molecule to goes into the cell. Yeah. And then after the DNA fragments are now inside the cytoplasm of the cell, the DNA fragment can be incorporated into the chromosomes. Yeah. Usually it is occur not in random, but they have specific site where this recombination can occur, but that's for higher level discussion. For now, what you need to know is just the general picture of how transformation in bacteria can occur. Yeah. Nah, what is the structure of DNA? Yeah. The secrets of DNA function and therefore of heredity itself are found in the three-dimensional structure of the DNA molecule. You already learned about math. Yeah. So the geometry of this molecule itself play an important role of how it can conduct its role, its function. Yeah. What is the structure of DNA? We will delve further. First, DNA is composed of four nucleotides. What is nucleotides? Nucleotides is the small subunits that compose the chain of DNA, where each nucleotide has three main components. Yeah, there are three. The first one, phosphate group. The second one, deoxyribose sugar. The third one is the nitrogenous base. Now, in a certain nucleotide, the nitrogen-containing bases, nitrogenous base, yeah, it can be thymine or T, yeah, the symbol is T, or it can be cytosine, the symbol is C, adenine or guanine. Adenine is A, guanine is G. Yeah. Nah, DNA is a double helix of two nucleotide strands. Yeah, so basically it is a double helix molecule, and hydrogen bonds between complementary bases hold two DNA strands together. So there are a lot of type of chemical bond in chemistry. You have covalent bonds, you have ionic interaction, and you have one of it is hydrogen bonds. In DNA, remember, not the covalent, not the ionic, but hydrogen bonds. Yeah, so hydrogen bonds is one type of bond, yeah, that is strong enough, but at the same time, It allows the biological system to have the dance of life, to have the flexibility that we will not have if we choose the other type of bond. Yeah. So the hydrogen bond is pretty unique and it is quite common in biology, in life system. Yeah. Not only in DNA, but later on in protein, we also can encounter this type of bond, hydrogen bond. Okay. Now this one, DNA nucleotides, there are four types. Yeah. You have phosphate. Yeah. Phosphate is the yellow circle. And then you have the pentose sugar, yeah, or previously just called as sugar, depicted as the blue pentagon. So the blue pentagon here is the pentose sugar. Pentose, penta means five. So that's why it is depicted in pentagon, not in hexagon, octagon, or other type, but pentagon. Yeah. Because there are five carbon atoms, only five carbon atoms in the sugar. It has 5C. Yeah, that's why we call it as pentose sugar. Yeah. And then the last one, you have the third component, which is called as the nitrogenous base. If the zoom is up, then you can go to the same link. Yeah, I'll wait you again in this same link. Okay, this the last one is the